The nice thing about metabolomics is that it is this mix of metabolites or components and the interaction between them that actually confers the trait on the fruit. So as I said, it's different, so it's completely complementary to the gene-targeted approach. So using a combination of the two, we have the potential to fully exploit the mutant population, hopefully leading to discovery of radically new and different fruit qualities. So I've just got a couple more slides. This one is just illustrating a pilot, fairly small-scale project that we've carried out, where we've screened 100 of our tomato mutant plants using a metabolomics fingerprinting technique called NMR. This essentially shows a fingerprint of the chemical composition of each of the distinct genetic lines. On the little graph on the right, you can see just a small portion of this NMR fingerprint. You can zoom in on different regions, depending on which classes of compounds you're interested in. And then essentially, because you end up with 100 different fingerprints, you resolve the data by carrying out principal component analysis. And this is shown on the graph on the left-hand side, where you can see that the vast majority of plants are clustering in the middle of the score plot. But we can identify some individual plants which appear to stand out from the population. And because we know as well which kind of fingerprints are associated with which chemical compositions, we know which direction we want to move on in the score plot if we want to achieve a particular change in composition. So using this approach to screen just 100 of the lines, we have been able to identify several metabolic outliers with potentially altered taste characteristics. So I just want to finally wrap up by acknowledging some of my colleagues and co-workers that have been involved in the work that I've touched on today. On the left-hand side, my colleagues from Unilever, in italics, some of my predecessors who are no longer working on the project, and then the team that we're very lucky to work with from Newnham's on the right-hand side. So thank you very much for your attention.
Nadie, pues yo sí quiero realizar una pregunta y se la voy a realizar ya a Jacquie. Voy a hacerla en español. Unilever es una gran compañía de alimentación en el cual la calidad es fundamental en sus productos. Está trabajando en genética para mejorar parámetros de calidad. Hemos visto que cuando un genetista trabaja para parámetros agronómicos rápidamente casi se le da solución. Cuando trabajamos contra plagas, enfermedades, contra cobertura de planta, contra fruto más maduro. ¿Qué ocurre realmente desde el punto de vista genético con esos parámetros tecnológicos como puede ser que el BRICS estemos tan parados desde hace una serie de años? Sabemos que hay variedades salvajes, Parviflorum, etcétera, que tienen un BRICS de 8. ¿Qué ocurre para que, por ejemplo, en los últimos años vemos las listas de variedades y en 5.5, 5.6 somos incapaces de subir ese límite?
Yeah. I'm not sure I completely understood the question, but we are not really focusing on BRICS. When we focus on other traits, for example, aspects of taste, for sure the baseline is that it must not be at the expense of BRICS or yield. That's just the background given position. So when we see, for example, in the NMR experiment, an altered chemical fingerprint, the first thing we need to check is we haven't done anything undesirable. But we're not specifically focusing on increasing BRICS further, so I couldn't really comment on how achievable that could be.